Assalamu alaikum. My name is Qudsia and I am from the Department of BS Biotechnology. My roll number is 002. The topic of my presentation is the applications of NMR spectroscopy. NMR is a phenomena in which particular atomic nuclei respond to the applications of the certain magnetic fields by absorbing or emitting electromagnetic radiations. NMR has long been used by the organic chemist, biochemist, and physicist as an analytical tool for the study of the structures, conformations, interactions, and the dynamics of molecules of homogeneous liquids or solids. Applications of NMR apply in the many fields like biology, physics, biotechnology, biochemistry, etc. Proton NMR and the carbon 13 NMR are also the applications of NMR spectroscopy. These are the applications of NMR, proton NMR, carbon 13 NMR, applications of NMR in biotechnology and biochemistry, applications of NMR in biomedical research, applications of NMR in the drug discovery, and the last one is applications of NMR in physics. Now coming towards the next slide, that is proton NMR. Proton NMR is the applications of a nuclear magnetic resonance with respect to the hydrogen 1 nuclei within the molecules of a substance in order to determine the structures of its molecules. In sample where natural hydrogen is present, practically all the hydrogen consists of the isotope hydrogen 1. That means having one proton for a nucleus. Simple NMR spectra are recorded in solutions and solvent protons must not be allowed to interfere. Deuterated solvents, especially for use in NMR, are preferred. For example, deuterated water, deuterated acetone, deuterated methanol, deuterated dimethyl sulfoxide, and etc. However, the solvent without the hydrogen, such as the carbon tetrachloride or carbon disulfide, may also be used. Proton NMR is a powerful tool for the molecular structure characterization. The next is carbon 13 NMR. Carbon 13 NMR is also the applications of NMR spectroscopy with respect to the carbon. It is analogous to the proton NMR and allows the identification of carbon atoms in an organic molecule, just as the proton NMR identifies the hydrogen atom. As such, carbon 13 NMR is an important tool in chemical structure elucidation in organic chemistry. For example, the carbon 13 detects only the isotopes of carbon whose natural abundance is only 1.1% because the main carbon isotopes. Carbon 12 is not detectable by NMR since its nucleus has zero spin. The next is applications in the biotechnology. In biology, NMR is fundamental for determining and exploring the structures of the proteins. For example, enzymes and receptors. It has been used to elucidate the structures and functions of numerous biological components. More recent discoveries include determinations of the structures of the influenza virus proton transporters and the consequent development of molecules that block this transporter and stall infection by the virus. NMR plays a key role in the development of the vaccines and treatments for the range of diseases including HIV, influenza, tuberculosis, and cancer by allowing researchers to understand how the disease-causing agents function and to identify the potential drug targets. NMR also provides a powerful tool for studying the conformational changes in the proteins and chemical kinetic processes which enables the mode of actions of enzymes transporters proteins to be elucidated at the molecular level. Similarly, NMR is being used to characterize the transitions of the proteins in the brain and to investigate the influences of such changes on neurodegenerative diseases processes, such as Alzheimer's disease, Parkinson's disease, etc. Ultimately, such NMR studies may again lead to the development of tests and treatments for diseases in man. The non ionizing and non invasive NMR techniques are being increasingly applied to problems in medicines. There are two major approaches. The first one approach is an analytical technique using NMR spectroscopy for the identification and quantification of metabolites in cells, tissues, and the organs. In vivo, NMR spectroscopy provides a window to the metabolic states of different cells, tissues, and the organs. The second approach is the production of NMR images of any section of the body. Applications in the biomedical research. NMR can identify the molecules to probe molecular dynamics and study the molecular interactions. In biomedical research, it has been applied to the structures and functions determination of important biological molecules such as drug targets, study of drugs and the interaction with their targets, identification of abnormal metabolites as a biomarker for specific diseases, diagnosis of disorders by imaging particular tissues or organs. The structures of reactants and amino acid derived product of a novel biosynthetic pathways through hydrogen 1, carbon 13 and the phosphorus 31 NMR analysis. Now coming towards the next slide, that is the applications in drug discovery. NMR spectroscopy has been a technology of fundamental importance to the drug discovery and the development for the past few years, principally through its uses in the structural elucidations of leads, drugs, their synthetic intermediates, and the degradation products and impurities of those drugs molecules. NMR spectroscopy has been metabolic profiling or metabolomic studies of drug metabolism, drug safety, drug efficacy, and diseases, through studies of biological fluids such as urine and the plasma, representing the change in applications from the chemical to the biological sciences. Applications in chemistry. Nuclear magnetic resonance spectroscopy is widely used to determine the structures of the organic molecules in solutions and study the molecular physics and crystals as well as non-crystalline materials. NMR is also routinely used in advanced medical imaging techniques such as MRI. All isotopes that contain an odd number of protons or neutrons have an intrinsic nuclear magnetic movements and angular movements, in other words, a non-zero nuclear spin, while all nuclides with even numbers of both have a total spin of 0. The most commonly used nuclei are the hydrogen 1 and the carbon 13, although isotopes of many other elements can be studied by the high field NMR spectroscopy as well. The key feature of NMR is that the resonance frequency of a particular sample substance is usually directly proportional to the strength of the applied magnetic field. It is this feature that is exploited in imaging techniques. If a sample is placed in a non-uniform magnetic field, then the resonance frequencies of the sample's nuclei depends on where in the field are located. Since the resolutions of the imaging techniques depends on the magnitude of the magnetic field gradient, many efforts are made to develop increased gradient field strength. The last one slide is about the applications of NMR in the physics. NMR is used in physics to study the high pressure diffusion, and you know that the diffusion is the movement of the molecules from one region to another region. NMR is used in physics to study the liquid crystals, rigid solids and the membranes. And liquid crystal is the states of the matters which has the properties between those of the conventional liquids and those of the conventional solids. So that's all from my side. Thank you.